Very clear.
Thank you. I've been asked to talk on third wave and Delta plus variant. Okay, I always give a disclaimer. My information is based on scientific literature and not on social media posts, especially WhatsApp University. And I do not have a doctorate. I wish I had one from WhatsApp University. I have no conflict of interest to declare. I have no shares in any vaccine companies or any companies for that matter. I don't own any shares. I'll try to be factual and not prescriptive. What I'll do in the next 15 minutes to 20 minutes is talk to you on why did the second wave occur at such a high magnitude? Will there be a third wave? If so, when would it occur? What would the third wave look like? What is the Delta variant and why? What are its implications? Now, those who forget history are condemned to repeat it. Now, this is a very famous saying by George Santayana, a great philosopher, essayist, and poet and novelist of the 20th and 19th, 20th century. And he said this, and this is a graph I'm projecting of the Spanish flu. Graph was made more than 100 years ago. The y-axis is the number of patients who got the infection, and the x-axis represents the time. And you can see that the initial Spanish flu outbreak was very small, and after imposing certain restrictions, there was a lull, and then it appeared as a huge second wave. And again, after, mind you, those days there were no vaccines, and again, non-pharmaceutical interventions imposed, and then there was a third wave. So for those who say that we did not know a second wave was coming or a third wave is going to come, I think that is absolute denial. COVID is a respiratory virus. It spreads exactly like Spanish flu, and COVID also mutating. Influenza viruses were known to mutate. To sit back now and say that we will not have a third wave or we will not have waves beyond that, I don't think is correct. So I think we have to learn a lesson from history. Now, several pundits wrote off their COVID after. There were wonderful articles written in newspapers. Very few of us had warned the government way back in November. On the day I retired on November 30th, I said there would be a third wave and it would occur sometime in the beginning of 2021. Of course, I was criticized for it as a pessimist, but this is public health. When there is a susceptible pool in the population that is either not infected or vaccinated, that susceptible pool will get infected. It's a novel virus to humanity. The only way you get immunity is through infection or vaccination. Okay, we all know why the second wave occurred. I will just recapitulate for you because this is bearing for the third wave. There were many small super spreading events that occurred in major cities and towns, like small partying, small get togethers, and hostels, students coming from other places. So these, I call them as sparks. Now, several sparks lit up into a fire thanks to the lack of COVID appropriate behavior, people not wearing masks. In the name of economic activity, we opened up everything and there was too much of crowding. And to top all this, we had rallies and religious congregations and elections. So, a right mixture for the virus to cause an explosive wave. The cream on the cake, the icing on the cake was variants. There came from December onwards across the globe, there were viruses that became more infectious. We call them variants of concern. So all this added. As a result, we had a very steep wave, which nobody said they predicted, but it was writing was on the wall, I believe. It caught us unawares. It was huge. We were not prepared in terms of bed capacity or oxygenation or essential drugs. And we had different states peaking at different times. Delhi and Maharashtra were the first, followed by Gujarat, Maharashtra, Madhya Pradesh, Karnataka, then Uttar Pradesh, Telangana, Andhra, and so on. And Kerala is still continuing to simmer. That's a very important issue you should keep in mind. Even today, they are above 10,000 cases. So, as a result of all this, systems responded late. So, what is the road ahead? The road ahead, this is one of my favorite slides, is on the left. It's a road with many humps or speed breakers. Why do you have these humps? Because, as I said, this is a novel virus. You get either immunity either through infection or vaccination. Till the entire globe, 80 to 85% to 90%. Last year, we used to say 60% is herd immunity. I believe that is no longer valid because of the emergence of variants. So you can get reinfections. So I think the bar now is around 80% to say that transmissions will come down in small waves. What we are having now is three wave patterns on the right. These are the possibilities for us. The first graph on the top, all waves are equal. That doesn't apply to India. The second graph in the middle, second wave was huge and third wave is small. That's wishful thinking, in my opinion. The third wave, the third pattern below looks little more realistic, but the third wave, the height of the third wave will depend on several factors. Is this really going to happen? Well, there are very reliable modelers in India, and one of the IIT Kharagpur has been doing a great job. I think it's IIT Kanpur is doing a great job, and they have been bang on as far as their predictions go, based on the data. The key data that is used is 7-day moving average. I'm sure all of you know that. It is the number of samples positive by the number of samples tested in the preceding 7 days. Now you plot it every day. That means if the 7-day window keep moving, and then you plot the number of cases and look at the average, and then you do modeling. Now the IIT model, we are today on July 1st. As you can see, we are in the receding phase of second wave. It is still simmering in some states. If everything is normal with virus mutations, then you will have a very steep third wave, is the prediction. And when will it be? It will be in July. We'll come to that why. If there are very strict interventions, and what are those we'll come to shortly, then you will have a much shallower wave. But if you're back to normal, then you will have an intermediate kind of wave. Now, a lot of this, unlike the first wave to second wave, predictions are more complex now because availability of vaccine is there. Between first and second wave, vaccine was not widely distributed and available. Now remember, vaccine is one of the biggest confounding variables that will impact the wave. And secondly is, of course, the way people behave. Generally, waves are spaced 3 to 5 months apart. Why does it have? Why does a wave rise and why does it fall? Wave rises because there are small super spreading events. They are sparks and they light up a fire and cases go up. What happens in the peak of a wave is when all governments and societies impose restrictions on people movement, which we call lockdown. A very extreme step. Had this been done at smaller levels, whenever there was a super spreading event, we had locked down smaller geographic areas, housing societies, wards in municipal areas, villages or taluks. We would not need to go to a national complete lockdown or a state level lockdown. So this wave lasts usually between 6 to 8 weeks and then it comes down. It comes down because human to human interaction is minimized because of lockdowns. And number two, It continues there because people realize during lockdowns that this is serious business, and they start wearing masks, and there is a certain amount of discipline in the way people behave. Then, after 1 to 2 months, human memory fades. People get back in the name of economic activity to crowding events, closed spaces. Then again, the virus starts super spreading events. The cycle repeats. This will happen till people 90 to 90% of the people are vaccinated. I was one of the first in the country way back in January to say that children maybe, and of course I was misquoted, maybe targets in the third wave because there's no vaccine for children. And we have seen between the first wave and second wave, the proportion of children getting infected is more. However, this is not to alarm people because children generally have milder infections. A very small proportion develop serious infections and MISC, which is a complication. So, what should we be doing? Of course, the most important is to emphasize on COVID appropriate behavior, wearing masks and maintaining social distance and hygiene. This is more easily said than done. Remember, I worked in EMHANS for 35 years, and I know that we know more about the virus and the disease and the mutations than about human behavior. Behaviors do not change with knowledge, behaviors do not change with force. Behaviors only change by providing alternatives. Fortunately for us, the alternative we have for not wearing mask or letting mask down sometimes or not maintaining social distance is vaccination. But then vaccination takes time, and it's going to uh, it's a very lengthy process, and availability is the bottleneck current. But most importantly, we should continue to impress on policymakers, administrators, and politicians to avoid. We know very well closed spaces and crowds are where the maximum spread occurs. We should continue to be prepared for a third wave in adults, all those adults who are not vaccinated, and this proportion, because plus post 45 the coverage of vaccine is better. It's about 30% to 40% today, at least one dose. The age group between 18 to 45 is going to be more vulnerable for getting the infection. Of course, we should have beds ready. Should we have a similar level of number of cases, essential drugs and oxygen should continue. And we should start preparing at least for getting infrastructure ready at district level for children in terms of some ICUs, some ventilators, and training. Schools, colleges opening. I think I'm not an education expert. I think that debate should occur now. Whether there is going to be full opening, there is going to be interrupted online, offline, or it's going to be only online. These are things that I think we have to do very cautiously. And of course, we should be monitoring the test positivity rate, and proportion of students who attend institutions should be tested. So this is what the government of India has said. And this tells you that whatever I told you of how to blunt the third wave is summarized in this one slide. Non-pharmacological interventions are very important. COVID appropriate behavior, and of course, increasing vaccination. The what vaccination we did on June 21st is phenomenal. We could do it. We
Hello.
did almost 85 lakh
Namaskar,
vaccines
namaskar, भैया.
given in a dose. But to sustain that is a challenge because supplies. And secondly, what will happen with increasing vaccination is after some time, people will not come to facilities. There are a proportion of people, at least 30%, to whom you have to take the vaccine because they don't have the time, the resources, or the knowledge and the ability to come and waste one day for vaccination. And there are people living in inaccessible areas. So we should now start thinking about how to take vaccination to people and not ask people to come to vaccination. And then, of course, genetic sequencing is very, very important because that is the tool to monitor emergence of a new variant of the virus. So I will switch tracks at this point and move on to the Delta Plus with a brief introduction on what is happening. You all know that coronavirus is an RNA virus and it has an enzyme called RNA dependent RNA polymerase. This enzyme makes mistakes when copying the genetic material. Why does it make mistakes? Because it lacks proofreading ability. So, to give you an example, there are 30,000 nucleotides in RNA of coronavirus. When it replicates, the chances are that one nucleotide will be different from the parent strand, which means 29,999 nucleotides are identical. So imagine the number of viruses that replicates in a human being. These mistakes keep accumulating. We call them mutations. And if they are in critical areas, they will change the structure, the chemistry of the virus in terms of amino acid, which in turn will change the structure of the virus and make it either more infectious or less infectious. The less infectious will fall by the wayside because that is evolutionary principle, survival of the fittest. The more infectious will replace what is circulating earlier. There are too many technical terms that were used. The Pango lineage, as it's called, calling viruses as A, B, C, D, and then within A, B, and C, they were B.1, B.2, and within B.1, there was subclassification, getting too complicated. So the WHO about a month and a half back gave these new variants, alpha, beta, gamma, and delta. Let me tell you the mother of all mutations actually occurred sometime last year, end of February, March. It was detected in Denmark, in Italy actually. And this grandmother mutant, as I call it, was a very significant change in amino acid in the spike protein at position 614 from aspartic acid to glycine. Now that changed the structure of the spike protein and made the virus more infectious. And as the graph here shows you, by May end and by June, July, entire globe was replaced with this particular mutant, which we call D614G. Now, this replaced the existing Wuhan virus at that time. So, this is the grandmother backbone on which all mutations are occurring. So, in late November, UK noticed a huge spurt of cases in Kent. And when they sequenced, they found that it is a new variant. And that was called UK variant. Now it's called Alpha. Then in December, South Africa reported one more mutation over the UK variant. And that was called the South African variant, now called Beta. And then, of course, in January, the Gamma was reported from Brazil and Delta was reported from India. We reported in February, but when we backtracked, the samples were from November of last year. So these are the mutations. For those of you, I won't burden you with the details of with the changes in amino acids. Plus, also remember, apart from mutations, certain portions of the genome have been deleted. The significance of these deletions, we are still trying to understand. But these mutations and change in amino acids, especially on the spike protein, makes the virus more fit. You know, viruses interact with ACE2 receptors. So if there is a key mutation in the spike protein, it engages better with the receptor, the fitness improves, thereby infectiousness improves. India set up in January Indian SARS-CoV-2 Genomics Consortium, a consortium of 10 labs. This is being set up by Ministry of Health and Department of Biotechnology, and there are till yesterday there were 10 labs. From today, another 18 labs have been added. To date, it has sequenced more than 45,000 samples from across the country, and the biggest contribution of INSACOG has been the detection of Delta variant. For the first time globally, we contributed to the detection, and also recently the Delta plus variant, and now there is a system that is put in place for sequencing. A word about sequencing: the purpose of INSACOG is to ascertain the emergence of new variants, to establish sentinel surveillance, and to look at trends of these variants. Basically, the samples collected from international travelers and other patients, I'll show you in the next slide, are sent, the VTMs are sent to the labs where the RNA is extracted. And then if the CT value is less than 25, it is suitable for sequencing. And these sequencing steps are: first we convert the RNA into cDNA, then we amplify bits and pieces of the DNA using a number of primers, that's called library preparation. Then we put it on a sequencing machine that breaks the code of DNA, and then that takes about all these takes about 3 to 4 days, and then it goes into a information pipeline where analysis is done, massive amount of data that takes 4 to 5 days. In the end of it, it generates a sequence, and that is compared with the existing sequences, and if it is a change, we call it a variant. That's called variant call. So the turnaround time for INSACOG is 15 days from the day the sample is received. Now the government of India has established across the country sentinel surveillance based on the WHO recommendation. Every state has 10 sentinel sites. 5 of these are hospitals and 5 of these are genomic labs. From each of these sentinel sites, every 15 days, 15 samples are collected. That means if there are 10 sentinel sites in a state, 150 samples are sent every 15 days to a sequencing lab. Sequencing labs have been mapped to states. And also they are now being increased. What are these categories of these samples? International travelers, because we can always import variants; representative samples from clusters and focal outbreaks; cases of COVID-19 reinfection and those with different clinical phenotype, more morbidity or mortality; infections, breakthrough infections, that is infections occurring in individuals after 2 weeks after 2 doses of vaccination; and of course, pediatric COVID-19 infections. Now, what India has done now is, this is a flavor for you. Week by week, we look at the emergence of variants. The color coding you can see. The indigo blue was a UK variant that dominated in January and February. Then we had a Delta variant, not the one that caused the huge outbreak. This was a variant that dominated in March and February. And then, of course, the Delta variant 6.1.7.2 emerged, was detected in Maharashtra in early March, and now it is completely taken over. And that is the pink or the mauve color you see. Now, most of the places across India are Delta plus variant. There are two terms you would have heard: variant of concern and variant of interest. Variants of concern means there is evidence both in the field that the number of people getting infected from the variant is more compared to, basically the R naught is more for these. And the second evidence is in the laboratory, people have grown the virus and shown that it multiplies more effectively than the previous. Variant of interest is, it is noticed in some clusters. It does spread, but it is not completely taking over a lineage. So it is under investigation. So it's called variant of interest. Coming to Delta plus. Now, Delta plus is a virus that belongs to the Delta lineage. The delta lineage itself has certain characteristic mutations by which it is called delta plus, and all of them are, these signature mutations are in the spike protein. Delta is characterized by two important mutations, and those two important mutations I did not show you earlier, but I'll quickly show you what they are. The delta has two important mutations. Remember, it is a derivative of D614G, the grandmother mutation, and then it has an amino acid lysine to arginine change at 452 position. So that is delta for you. Now on delta, there was another mutation, and this mutation in delta was in a position at 417. Now, this is a change from lysine to arginine. Now, this K147, K417N, interestingly, is a mutation that was present in South African variant. Now, Delta Plus has acquired that mutation. Now, there are in virology circles people now talking about recombination. We never knew coronaviruses can recombine. There are evidences in animals. So is it possible that South African variant and Delta variant infected the same individual and within his cells there was recombination and this was produced? We do not know. Because it has this K417N, it is worrying people. Why is it worrying people? Because K417N in South African variant is one of the mutations that are associated with vaccine escape. Remember, all the vaccines that have been licensed for human use now are effective against all variants. What do you mean by effective? They prevent severe disease, they prevent mortality, but they don't prevent infections. And it has been noticed among all the variants that the beta variant or the South African variant is the most resistant to most of the vaccines. Now, here is Delta Plus that has acquired one mutation from South African variant and put it into Delta. Consequently, the evidence we have today is that it escapes treatment with monoclonal antibodies. That makes a lot of sense because monoclonal antibodies are directed to a particular domain of spike protein known as receptor binding domain. So this mutation is in the receptor binding domain. Therefore, the mutation is rendering the virus resistant to monoclonal. We have reported around 52 cases today in 10 different states. Maharashtra has reported around 27 odd, and there are other states, and totally 20 countries have been reported this. Now, the most important questions being asked is where did this lineage was first detected? We don't know where it originated. It was first reported Public Health England in 63 genomes in about the first week of June. But actually, these mutations have been noted in samples collected much earlier. In the global database, it was detected in first week of June, and England flagged it up. And we also went back and we also found that we've had Delta Plus variant in our country from middle of April. Is it more infectious? The answer is there is no data to say it's more infectious. The R naughts are being worked out, but common sense tells you that if it was more infectious, we are talking of Delta Plus in India in middle of April, and we are now beginning of July. It's a clear 2.5 months. Had it been more infectious, and had it been R naughts or more, one would have expected sweeping outbreaks by now. The only reason is we were very careful. We were in lockdown, so maybe it's not occurring. But as of now, there is no data. It's being grown in the lab, and they are looking at its replication efficacy and comparing it to Delta and other variants. My own gut feeling is it is not more infectious. Is it more virulent? It doesn't look so. If you look at the clinical presentations of Delta Plus, it is no different from Delta. It is doing whatever Delta is doing. It is not killing more people. So I don't think it is virulent. Will current vaccines work against Delta plus variant? What do you mean by work? When you say protection against severe disease and death, it will, all vaccines will work. When you talk about breakthrough infections, it may have more breakthrough infections, because remember, it has one key mutation which it has acquired from the South African variant, which was the resistant one. So should we be unduly concerned, worried, panic? Concerned, yes. Worried, yes. But there's no cause of panic, because, as I said, it should have caused a lot of infections by now. To summarize, then, SARS-CoV is a new pathogen, contagious, spreads quickly. If you're not vaccinated or not infected, the virus will catch you. Especially if newer variants emerge, there is a chance of reinfection. But immunity will, in reinfection, will protect you from serious disease and death. The road ahead is bumpy. We should learn to apply brakes, like when we cross a speed breaker, and do continuous monitoring of test positivity rate, apply local clampdowns and local isolations to avoid a lockdown, total lockdown. Delta Plus is a new variant, and it is a variant to watch. No evidence to suggest it is spreading more, or it's more infectious, more virulent, or as a matter of concern. Thank you. I'll stop here.